Good day, we are Group 2 from Section D, and we are going to talk about pyridoxine or vitamin B6. Vitamin B6 is a water-soluble vitamin that was first isolated in 1930s. It is naturally present in many food, added to others, and available as a dietary supplement. Pyridoxine occurs primarily in plants, whereas pyridoxal and pyridoxamine are found in animals. B6 is absorbed in the small intestine, particularly in the jejunum, metabolized to active form in the liver, and excreted in the urine from the kidney. Pertaining to its cellular structure, vitamin B6 is the collective term for a group of three related compounds: pyridoxine, pyridoxal, and pyridoxamine. While all three of these variants can be phosphorylated, the phosphate ester derivative of pyridoxal, which is pyridoxal 5-phosphate or PLP, is the cofactor of most vitamin B6 dependent enzymes in the body. These three differ only in the nature of the functional group attached to the ring: alcohol for pyridoxal or pyridoxine, aldehyde for pyridoxal, and amine for pyridoxamine. Pyridoxal phosphate is a coenzyme for many enzymes involved in amino acid metabolism, especially transamination and decarboxylation. For example, the transamination of oxaloacetate and glutamate will yield aspartate and alpha-ketoglutarate. Also, the deamination of serine will yield pyruvate and ammonia in the process, but the decarboxylation of histidine will result to histamine and carbon dioxide. Finally, the condensation of glycine and succinyl-CoA will yield delta-aminolevulinic acid. Vitamin B6 is also the cofactor of glycogen phosphorylase, whereby the phosphate group is catalytically important. Moreover, it is important in steroid hormone action, in which pyridoxal phosphate removes the hormone receptor complex from DNA binding, terminating the action of the hormones. PLP-dependent enzymes are involved in essential biological processes, hemoglobin and amino acid biosynthesis, as well as fatty acid metabolism. PLP functions as a coenzyme of 5-aminolevulinic acid synthase, which is involved in the synthesis of heme, an iron-containing component of hemoglobin. PLP is a coenzyme of reactions that generate glucose from amino acids, a process known as gluconeogenesis.
Next is the biochemical pathophysiology of clinical manifestation. The vitamin B6 deficiency. Isolated vitamin B6 deficiency is happened because of the low concentration of other B complex vitamins such as vitamin B12 and folic acid. The clinical deficiency diseases are rare, but the significant proportion of population have a marginal vitamin B6 status. The deficiency results in abnormalities of tryptophan and methionine metabolism. Next is, it is caused by dietary malnutrition, alcoholism, pregnancy, and certain metabolic diseases, and certain pharmacologic agents that interfere with the pyridoxine metabolism or act as competitive inhibitors at the pyridoxine binding sites. Next is the clinical features that are neurologic, integumentary, hematologic, and psychiatric. Groups at risk of vitamin B6 inadequacy, they are the individuals with impaired renal function, individuals with autoimmune disorders, people with alcohol dependence. Next is, why would a vitamin B6 deficiency result in small microcytic pale hypochromic RBCs? In vitamin B6 deficiency, the rate of heme production is low because the first reaction in heme synthesis requires pyridoxal phosphate. Thus, less heme is synthesized, causing RBC to be small and pale. Iron stores are usually elevated. This is the treatment shown here. Next is the toxicity of pyridoxine. The development of sensory neuropathy have been reported in patients taking 2 to 7 pyridoxine per day for a variety of reasons. There are some residual damage after withdrawal of these high doses. Other reports suggest that intake in excess of 100 to 200 mg per day are associated with neurological damage. Substantial improvement but not complete recovery occurs when the vitamin is discontinued. Vitamin B6 status is assessed by assaying erythrocyte transaminases. The most widely used method of assessing vitamin B6 status is by activation of erythrocyte transaminases by pyridoxal phosphate added in vitro, expressed as an activation coefficient. Measurement of plasma concentration of vitamin are also used.